Let's take a look at some factors that affect the ability of an enzyme to do its job. So here, these two graphs show us that enzyme can be affected by temperature and pH. And some enzymes are temperature specific, as in the first graph here, this human enzyme being active at human body temperature, 37 degrees Celsius. Here we have an enzyme that would be in the cell of a thermophile bacteria, okay, that this hot springs kind of bacteria would have enzymes that would function at a much higher temperature. Similarly, pH has that kind of effect. Here we have pepsin, which is the protein digesting enzyme in your stomach, is optimal at a pH of about 2, 2.5, whereas trypsin, okay, which is going to work in your small intestine, has an optimal pH range of 6.5, getting closer to 7. So how does pH exactly do this? Okay, so here we have three different enzymes. Pepsin, which works at a very low pH. Salivary amylase, which is the saliva enzyme that starts to break down carbohydrates in your mouth. Okay, and arginase, okay, which is going to work at a much higher pH. Arginase breaks down the amino acid arginine into these two amino acids, okay, into this amino acid and urea, pardon me. Okay, the ornithine, okay, it's the start of a very important factor in the start of the urea cycle. Okay, so we see here is the enzyme working on arginine, breaking that down into urea and ornithine, and continuing on around the urea cycle. So this is going to be much more active. So this will be active in the stomach, this will be active in the mouth, and the arginase producing urea would be very active in the kidneys. So let's take a look at other important factors that affect our enzymes. Okay. Other important factors include cofactors, coenzymes, and inhibitors. And let's take a look at those. Cofactors and coenzymes are quite similar, except that coenzymes are organic cofactors, where cofactors are inorganic. Okay, let's take a look at what those are. Okay. So here we have an enzyme who's got to work on this hexagon-shaped substrate, but the active site isn't quite ready for that substrate until the cofactor or coenzyme first connects to that active site, making it the proper shape, and now the substrate can fit in that active site, and we can have the reaction catalyzed from there. So the cofactor helps the enzyme take on the proper shape. Next, we have inhibitors. Inhibitors can either be competitive or non-competitive. So whereas the cofactor or coenzyme helps to maintain the active site shape and encourages the enzyme to work, now the inhibitor interacts with the enzyme in such a way as to discourage its function. Okay? So the competitive inhibitor actually bonds to the enzyme's active site and competes with the substrate for the attention and bonding of the active site. Okay? A non-competitive inhibitor bonds to the enzyme somewhere other than the active site, but in doing so changes the conformation of that enzyme in such a way that it can no longer bond with its substrate. All of these are ways that Mother Nature controls enzymes: temperature, pH, cofactors, coenzymes, and inhibitors, so that we can have these chemicals interacting with substrates when and where we need them to. And I hope that's helpful.